And now I'm going to talk about the differences between genotype versus karyotype versus phenotype. All right, so again, let's go back to the cell. So the cell has 46 chromosomes or 23 pairs. And when we're looking at the genotype, and within each chromosome, remember we have these alleles here. These are called alleles that come from the mom and the dad. So we have big B, little b, big B, little b, which determines, for example, what color eye the child is going to have. Although we have had, my cousin actually has a blue and a green eye. It's kind of pretty, actually. So, but normally that is what's supposed to determine it. So these are the alleles right here, and the genotype is what refers to is the pairs of alleles. So that will be just talking about these specific pairs. That's the genotype, whereas the karyotype is actually looking at the entire cell of all of the chromosomes put together. So when I think of it as taking a photo of this cell over here with all the chromosomes. That's the karyotype, whereas the genotype is digging down into these alleles right here. Whereas the phenotype is not necessarily paying attention to the specific alleles, but it's looking more at the characteristics. So what develops from these, from the genotype and the characteristics. So we then look at behavior, we look at appearance of the person. So that's the phenotype. So when I think of that, I think of appearance, behavior, certain characteristics, or all characteristics. Karyotype is a photo of the entire cell making up all the chromosomes. And the genotype is talking about each allele as it explains, BB, BB, and little b. The difference between monosomy and trisomy. Monosomy, remember that there's 23 pairs of chromosomes, and sometimes we can have a missing chromosome or an extra or a chromosomal pair. They don't separate and they remain together in the one sperm. And so then what happens is an extra chromosome can join them from the egg of the female. And that would be an extra one. So when we think of pairs, that's how it usually develops. But sometimes it'll end up doing an extra one because these two stick together, the sperm, and then we have the egg brings this one, an extra one in, and now we have three. So for example, trisomy 21 means on number 21 chromosome, there is an extra one. So that's what trisomy 21 is. And there's different trisomy disorders. And depending on the disorder will depend on the person's symptoms and characteristics that they end up having. When we do the monosomy, that just means that instead of the pairs, one from the man, one from the woman, one is missing. So that's monosomy. So and again, when you're describing or when you hear these disorders, if you say number 46 monosomy, that just means it's not a pair, it's absent. And then they will display characteristics, developmental issues, depending on what exactly the characteristics are of that specific chromosome. When we talk about meiosis and mitosis, that is dealing with cell division before we even get to chromosomes. So when we think of meiosis, it's the division of the sex cells, or germ cells is another term for it. And mitosis is body cell division. So they are two different things. When you think of meiosis, you're thinking of sex cells or the germ cells, and the mitosis is body cell division. The other difference is the meiosis, since it's the sex cell or germ cell, it's going to divide twice. And whereas the mitosis, it's only dividing once. So we talked about before extra chromosome or missing chromosome, and that has to do with a numerical issue. So we talked about that for the example, the trisomy 21, or in someone lacks a chromosome, it can be called Turner syndrome is another one. And that means a female is born with only one sex chromosome, which is the X, and is usually shorter than average and unable to have children and other issues. Again, remember, it depends on the chromosome. So we talked about numerical abnormalities, but now let's talk about structural abnormalities. So make sure you review this, but I just want to do a quick go over with you. And this site here is from the genome.gov. And so if you want to go to that, it gives some really good information. And here's a picture here from the NIH also about chromosome abnormalities and pictures. So here we're talking about structural abnormalities and not necessarily the numerical abnormalities. So when we talk about deletions, all right, so we'll think about this as a chromosome. Part of it is missing or deleted. And again, it will depend on what's missing to the specific type of characteristics that the person will have when they're born, and all the multiple issues. When we think of duplications, part of this over here, duplications, it's duplicated. So that means there's some extra genetic material, which can cause some developmental issues. Translocation means where a portion of one of them is transferred, the portion of one of the chromosomes, so say this little area here, it's transferred to the other chromosome. An inversion, that just means a part of it has broken off, turned upside down. So when you think of inversion, just think it turned upside down and then reattaches itself. And then rings, which, it's not drawing on here. Oh, it's because I have this. So rings is the portion that has broken off and formed a circle or ring around it. And sometimes it can lose genetic material, but sometimes it can stay intact. So one of the most important ones, or actually the important ones I want you to make sure you understand, though, is the translocation. And also I want you to look up non-disjunction. And what that means. So in particular, Klinefelter syndrome is one example. So look up Klinefelter syndrome and understand why that is a non-disjunction issue and how does that happen. Also, I'd like you to look up what does inactivation of a chromosome mean. And you know translocation, so make sure you understand, is it erasing? Understand all of these, the deletion, the differences between them. And when you think of those of you that are visual, can look at this picture over here and understand what they are. But definitely go a little bit further and figure out, try to understand also what a non-disjunction really is. The other thing that is not something that you have to know for exam purposes, but it's very interesting because why does this happen? Why do we have deletions, duplications, and just specific gene disorders? Well, obviously there's still always research going on, but what they have found is that the females, we only have a specific number of eggs that we're going to have, and we have those for the longevity of before we are postmenopausal. So there's research out there that our eggs can age basically, and they can go through sometimes have some genetic dysfunction to it. So that's why if you're over 35, that's why you're considered more high risk, because you have a higher risk of the egg not necessary, or maybe not producing correctly or dividing correctly. Whereas the male, they don't think paternal age has anything to do with it, because males are continuously producing new sperm every time, whereas the females aren't. And lastly, I want to talk about the different types of disorders. So there's autosomal dominant, autosomal recessive, there's X-linked, dominant and recessive. There's also other ones on the side here. So that this is in the table in the table. Make sure you understand those. There's also Y-linked, there's mitochondrial issues also, but I want to make sure that you specifically focus on these and understand what they are. And also, I know that Professor Shaw, or now Warzak, went over the table with you to try to help you determine whether someone will be, if there's a 25% chance of the child having disease versus 50%. So make sure you review that. Also understand what the X-link is, and let's just say an X-link is in the family, so who is more likely to inherit? Would it be like a son or a daughter, etc.? So make sure you understand those.